So this talk today, and it's about doubting the neurofeedback double blind. Again, thank you for inviting me and letting me present. The main question I'll be asking is if participants have residual awareness during experimental purposes in neurofeedback studies, whether participants can know that they are being trained in neurofeedback studies. Do they have some kind of residual awareness? And to get started, neurofeedback is a psychophysiological procedure in which online feedback of neural activation is provided to the participant for the purpose of self-regulation. So it's basically a type of reinforcement learning where you gain control over your own brain. So you have this subject inside a scanner. It could be an MRI scanner or EEG. And then we pick up a signal, a data signal from that brain, and then we analyze it rather quickly and show it back as a feedback signal to the participant. And here you can see it's kind of set up as this target level that the participant has to reach. So it is then a type of game, you could say, where the participant learns to control their own brain activity. It is fed back to the subject, and that affects repeating measures of this loop here. And so each time the participant learns to gain control over their own brain, they in a way get a reward. And that allows us to train participants into controlling brain regions or different types of brain activity. And we know the brain is involved in all kinds of fascinating cognitive functions like attention, perception, motivation, intelligence, and so on. So that allows us to train those types of cognitive functions. But it also allows us to train clinical aspects that can be measured in the brain, like pain, for example, which is useful for all of medicine. It could be measuring cognitive decline or symptoms of cognitive decline in the brain, which is useful for neurological patients, or it could be measuring symptoms of depression and anxiety for psychiatric patients. So there's potential here to have clinical effects. Some of the sort of historical roots of neurofeedback was by Joe Kamiya, who in the 60s did the first neurofeedback study. Joe Kamiya was a Japanese American who first showed what he called conditioned discrimination of the EEG algorithm in humans. So what Kamiya did was he waited for 2 to 6 seconds of alpha activity. This is a type of brain activity, or non-alpha activity, so when there was high alpha or there was low alpha, and then he prompted participants to answer if they thought that they had high alpha or low alpha. And it turned out that participants could learn to discriminate their own brain activity. They could learn to know whether or not they were in a high alpha state or low alpha state. And so 9 of 12 subjects, they reached a significant proportion of correct discrimination after 7 one-hour sessions. So they were able to figure out whether or not they were in an alpha state. So this is conditioned discrimination. But this can also be, this type of neurofeedback can also be extended to sort of clinical aspects where you can train certain brain areas specifically. Now we have fMRI, for example, where we can measure brain activity in different areas. And one such area is the anterior cingulate cortex, which we know is involved in pain perception. So research, for example, within hypnosis has sort of looked at if you ask people to catastrophize about pain, if you ask them to feel a pain as more painful, it activates the anterior cingulate cortex. So we can then use neurofeedback to downregulate the activity in anterior cingulate cortex for chronic pain patients. And that allows them to gain control over their own pain. So again, we're measuring the activity in this area, and each time the participant lowers this activity, they're given a reward. They're getting a positive feedback signal. And sort of the first study that showed this was Decoded in 2005. What critically is shown is when you compare this type of feedback, neurofeedback, to other types of control conditions where the participant just has to attend to their pain, or the activity from neurofeedback is not derived from the anterior cingulate cortex, but is derived from another area of the brain, in this case, the posterior cingulate cortex, or it is derived from another participant, meaning you are just getting false feedback, then you don't see a downregulation in pain. It is only when it is neurofeedback from this specific area that you see this effect. So that allows you to have control groups that can account for the placebo effect, because pain patients have a big expectation to relieve their pain, and they're really motivated to relieve their pain. And that is a kind of placebo response that you want to control for. And this is also the sort of consensus right now in neurofeedback studies, is that we need more double-blinded randomized controlled trials to make sure that these effects are not due to placebo. Neurofeedback, I find, is very interesting, because in comparison to traditional neuroimaging, where you just have a cognitive function that can change, it can go up and down, so pain can go up and pain can go down, and activity in some area goes up and goes down, like I mentioned before, this anterior cingulate cortex, that only allows us to infer a correlation between the brain activity and the cognitive function. But when we have neurofeedback, we're able to train this area and change it up and down, and see corresponding changes in the cognitive function going up and down. And this allows us, similar to brain stimulation, to do a type of causal inference, where we can say that because we changed the activity in that area, there was a change in that cognitive function. But one problem here, like I mentioned, is this placebo effect, which is, well, let's say that we do train this area of the brain, but it indirectly affects some other area, maybe a kind of placebo area of the brain. And this placebo area is actually the one that truly causes this cognitive function. Well, then we don't know, because we don't know if training that area is actually indirectly affecting other areas. So neurofeedback has this problem that we might be affecting something else, like placebo and so on. Yeah. So it's important in neurofeedback to handle expectancy. How much expectation do the participants have? So a good way to do this is to ask participants before they do a study, are you expecting to get relief from neurofeedback? How strong is your expectation? And then to ask participants if they think they were in the active control condition or in the target group or in the control condition, and to test if they actually know whether or not they were receiving the real neurofeedback or kind of false neurofeedback. And then to test if blinding was properly performed, if the participant is truly blinded to the study, so a double blind. This is also the case when you look at antidepressants, for example. This is a similar type of thing. And in comparisons to antidepressants, for example, for depression and anxiety, we know that the large amount of the effect is due to placebo. The strongest determinant of the effect from antidepressants is due to a placebo response and not the actual medicine itself. And in this regard, neurofeedback is showing stronger effects. It's showing that the effects we're seeing for depression and anxiety simply exceed what you would expect from placebo effects and other drug effects. So that's very encouraging for neurofeedback. But we still need something more. We still need some more tools to make sure that we ensure this double blind. And here, a new approach has been what is called decoded neurofeedback, or you could call it machine learning neurofeedback. And one of the seminal studies done by Shibata in 2011, which was published in Science, is sort of the first study to do this. And I'll explain this study in great detail, and it's one of the most important studies to address in this area. So what they used in this, what you can do is you can, if you present a stimulus to a participant, let's say a face like this, or a shoe, or a chair like this, it activates certain areas of the brain, certain specific areas of the brain. And you can use machine learning or decoding to figure out the exact voxels, in this case, the exact spots in the brain, the representations in the brain that are active for these different stimuli. And what Shibata did was do this for 3 different orientations of lines. So this line is at 10 degrees and 7 degrees and 130 degrees. And you saw the brain areas involved in this in these degrees. Then what he did was each time the participant reproduced these activities. For example, if a participant was put into it was to train the 10-degree representation, then every time the brain recreated this activity, this feedback signal that they were viewing during neurofeedback became larger. So in a sense, he was asking participants to recreate the activity in their brain from when they saw this 10-degree angle. And by replaying this again and again and again in the brain, just by you increasing this feedback circle, you are able to recreate the brain patterns from when you were seeing this 10-degree angle. And it's important to understand here that the participants are not actually viewing this degree. That's only done in the beginning, and then the neurofeedback trains them to recreate this pattern of activity while they are doing the neurofeedback. And critically, when the participants are tested afterwards, so in a test afterwards, they have to discriminate between these 3 different degrees. These degrees are presented in noise, and they're asked in like a 3 alternative forced choice tasks to discriminate these 3. They're much better at discriminating the degree that they were trained on. So they've recreated this brain activity and trained the brain to represent this degree to a better extent, and now they're better able to see that degree, even if it's masked in noise. So that allows us to, or that allowed Shibata to train very specific cognitive function, a very specific visual degree. And this is very, very interesting. This is actually very, very fascinating that we can train these types of cognitive functions like this. What also was part of this study was a claim that this was done without awareness, that this is done completely without the participants knowing. And because after the study, when Shibata and colleagues, they asked the participants what they thought, what kind of strategy they used, they said they had no idea what kind of strategy they were using. And therefore, they conclude that the participants didn't know what they were doing, and they didn't seem to know whether or not what they were training and what was happening. They were just increasing this feedback disk, and then they became better at discriminating these lines. And they didn't know what the purpose of the experiment was. So I want to make a parallel here and say that in consciousness studies in general, in consciousness research, we have a lot of claims of unawareness. So for example, we've had the claim that when you're dreaming, you're unaware that you're dreaming. This used to be a claim that was very much present, that when you're dreaming, you don't know that you're dreaming. But then we figured out ways to show that people can have lucid dreams, where people are actually able to, when they're lucid dreaming, to communicate via eye movements or EMG activity, where they can move their hands while they are dreaming and still have what's called rapid eye movement, EEG sleep patterns. So they're able to communicate that they are aware that they're dreaming. So we have this claim that in dreams you're unaware that you're dreaming, and then we find some better methods, and then we can show that you are actually aware. We also have had in consciousness a claim that patients in vegetative states are unaware. So patients who come from a coma and go into a vegetative state, they show signs of being alive, and they show signs of waking up, but they cannot communicate, and they're sort of in a vegetative state. They're not showing signs of awareness. Was the definition of vegetative state. But then we found a way to test in an MRI. If we ask them to reproduce a certain type of brain activity, we ask them to imagine playing tennis, then they show activity in the same area as controls. And if we ask them to navigate around their house, so imagine walking around your house, then they show activity in the areas of spatial navigation imagery similar to controls. So we had this claim of unawareness, and then we've developed better methods, and now we have a new understanding that there is actually awareness. In terms of visual perception, we once had this idea that if you have visual discrimination without explicit awareness, then it reveals subliminal perception. So this was a critical claim in the start 90s that if you, for example, present this stimuli to participants like a square like this, or it's a square like this, or it's a tilted square like this, and you present it very fast, so 33 milliseconds, which is very fast, and then you put a mask on top of it, so it's really hard to see. People, they don't report that they saw anything, but then you sort of ask them, okay, so did you see this square or that square? And participants can sometimes actually have above chance performance on discriminating the two, even though they say that they did not see anything. And this was referred to as subliminal perception, that you have discrimination, you have accuracy without explicit awareness of this this square here. So this was the claim, that there's this accuracy without awareness. But then it was developed this test scale, which instead asks people in terms of reporting in terms of no experience, weak glimpse, almost clear experience, or clear experience. And when we ask people to do just that, then we find that this awareness scale maps on much better to discrimination performance, and that this is actually what participants would choose to describe themselves. If you ask them themselves to sort of develop their own way of answering whether or not they saw it, they will actually say most likely, I didn't have an experience, or they will say I had a weak glimpse, or I had an almost clear experience. So this scale is much more intuitive for participants. And so again, we have some sort of claim of unawareness, and then we have a new method being developed that is more sensitive to awareness. And then we find that there's actually a degree of awareness, a residual degree of awareness, when we've developed better methods. And sort of to take the parallel back to neurofeedback, well, now we have this claim of neurofeedback without awareness. And I think it's time to ask if maybe participants do have some awareness of the experimental purposes in neurofeedback studies after all. And this is a question for consciousness research, I would argue. And it's a very important question because it has clinical implications for double-blinded RCTs that are being run using neurofeedback. And this is actually also the conclusion of the Nature Reviews Neuroscience paper by Sitaram in 2017, where they sort of the end of the systematic review, they say that we need more double-blinded RCTs of neurofeedback. It would be very good if we had a type of unaware neurofeedback, because then we could circumvent the problem of placebo effects. And this makes neurofeedback have greater amount of clinical strength, or a potential to overcome placebo effects. One study that I'll also mention, which is a new study that shows this exactly, is if you have people who are afraid of certain animals, for example, snakes, you show them snakes, and you show them butterflies. So you have this target stimuli, and you have this butterfly. What you can do is you can actually figure out the representations in their brain through those snakes. And one way of doing is actually not using their brain activity, but using another participant's brain activity, or a surrogate brain. So that allows you to, without even showing them the snake, to train those areas of the brain that are related to the perception of a snake, just from inferring it from other participants or surrogate participants. What you can then do is you train this activity of the brain of the target participant. You train those areas that you believe would be active if they saw a snake. And by changing this activation likelihood, again, the participant is just asked, increase this feedback disk. They're asked to make this larger. Then you can downregulate the area involved in the representation of snakes. And for this patient who is afraid of snakes, what you can then have is, before the neurofeedback, their amygdala activity. Amygdala is involved in fear processing and the explicit or implicit emotions. You see that their amygdala becomes very active when they see a snake. But after neurofeedback, they decrease the activity in the amygdala. And whereas the control condition does not change, and also the skin conductance response when they see the snake is very large when they see the snake the first time. And after this type of neurofeedback, this decoded neurofeedback, they don't have the skin conductance response, whereas the control group still have. So this is a very big clinical potential. And you could say, yeah, but what is happening here is the participant is just viewing this feedback disc, they're training this feedback disc, and they know that they're training something in their brain. They know that they're undergoing some sort of treatment. And you could say that, well, the participant is coming in, they're recruited for anxiety, they're recruited for some clinical purpose, depression, pain, and so on. And they know that they're part of the study because of that, and that's why they may know that they're undergoing some sort of treatment. But that's actually also a way to also circumvent that problem. So one study performed by Ramot in 2016 is what is called covert neurofeedback. So this is also claimed to be without awareness, and this is in a very smart way. So the participant is lying inside the scanner, and they hear sound. So they just hear some sounds that are playing, and they're told that some sounds, if they hear one certain sound, like a pling noise, something nice, they win money. And each time they hear this noise, they actually win money, and they will get real money when they come outside. And when they hear other types of sounds like dude, they lose money. And they're just asked to listen to these sounds. And sometimes participants, they really hope that they're going to hear the pling sound and win some money. What is actually happening without the knowledge of the participant is that they are measuring activity in two areas. And each time the activity in one area is increasing and the activity in another area is decreasing, they play this noise, this pling noise, this good noise. And when the brain does the opposite, when the other area becomes active, they play the noise that indicates that they lose money. So just because the participant is hoping for a particular type of sound, they're actually changing their own brain activity while they're sound. And each time they do, and they end up increasing the activity in the area, they get a reward. So in that way, you're training their brain without the participants actually knowing it. They have no idea that they are actually receiving this type of neurofeedback. What Ramot did in the study is at the end of the neurofeedback session, they asked the participants, do you think that we were trying to change this face-related imagery or this house-related imagery? So yeah, I should say that the two areas that they were training, the fusiform face area or the parahippocampal face area, the FFA is involved in face perception, and the parahippocampal place area is involved in place perception, or remembering certain places or houses, you could say. So they asked the participants if they think that the training was involved in these two types of cognitive functions, or they asked them decoy options. They said, maybe we were training these abstract visual forms or language or body motion related imagery, and the participants could not guess the correct things at the end of the study. So there has been, you could say that this, without the participants even knowing it, is a new type of neurofeedback called covert neurofeedback. And there's a nice review in Neuroscience and Biobehavioral Reviews that sort of rank the different types of implicit neurofeedback studies that sort of says that this is a new type of neurofeedback where the neurofeedback is done in a covert way, this Ramot study. So you can really have the presence of a feedback signal, or you can have this indication of was awareness there or not, and are they actively trying to control something? And if they are not actively trying to control something, if they're just lying there and listening to these sounds, then it's a different type of neurofeedback study. So it's also that we will dig into some details now. Importantly, they say that the participants were not informed of the fact that the reward dependent of their brain activity. Participants were told that the experiment was aimed at mapping positive and negative reward responses, and that they would receive money for these sounds. And then on 5 separate days, the participants were doing this inside the scanner. They were doing this type of neurofeedback. Finally, at the end of the study, it was revealed this fact that the 2 areas of the brain was involved, and that they were getting this feedback based on this brain activity. And then Ramot says that all participants reported having no notion what area was being trained, and what the feedback was related to. And then they were given this 5 alternate 4th choice task. And Ramot says that they were explained that which region or function it was associated with. So participants were allowed to choose between which region or function was associated with positive and negative rewards. Again, face-related imagery, place, house-related imagery, abstract visual forms, language, body motion-related imagery. And this resulted in guesses at chance level. So one thing I will say about this is that this seems to be very difficult for participants to know what brain area they were training. It's not likely that you as a person know when you are doing a type of training that you know which brain area is involved in that kind of training. At least someone who is not a neuroscientist will have a hard time saying, okay, I'm training my perception right now, or I'm training my face-related imagery right now. I mean, I try to make up an example of this. Let's say you were invited to do a repetitive TMS study. TMS is where we can activate certain areas of your brain with this magnetic pulse. And let's say that you are measured on a task. Let's say you're measured on a reaction time task where you have to press really fast on a button, and then the scientist puts a repetitive TMS coil over your premotor cortex and activates that area, and you know that he is activating that area, and then you're tested on a reaction time task again. Then you might be able to figure out and to know that okay, maybe the purpose of the experiment was to increase my reaction time or decrease my reaction time, and because you've been tested on reaction time task before and after, and because you maybe know like, the premotor cortex is involved in reaction time. That's something maybe a neuroscientist could do. In this case, these participants were not tested on a task before and after this type of feedback. They were just put inside the scanner. They were just listening to sounds and getting this feedback, but they didn't know. And then they were asked to relate that to brain areas and functions afterwards. And I would just argue that this is difficult for even a neuroscientist to do if there's no task before and after, and it's also going to be very difficult for a person off the street to say which brain area that was being trained. In the Shibata paper, regarding the claim that participants were unaware of the purpose of the training, they say that the participants were not able to guess which target orientation they were trained. So if they were trained on the 130 degree, then they couldn't guess it, and they guessed also the other degree. So participants were in general not able to guess the target degree that they were trained. Some participants did guess, but overall they were not able to guess it. So this, in a way, indicates that they're not able to guess which brain region that was being trained. They're not able to guess which cognitive function was being trained. Shibata and colleagues also mentioned that in the supplementary material they show that the participants had no true knowledge of the workings of the experiment. Here they asked participants what type of strategy they used to train to increase the size of the feedback disc. Participants are asked what their strategy was, and participants say that I tried to remember various scenes from one of my favorite animated movie, or I tried to switch my attention between two eyes, try to focus, and so on. But none of them were saying anything related to the orientation of certain lines that they were actually training. So this, in a way, is asking them about their strategy. And in my opinion, this is not asking them what they think the purpose of the experiment is. And it's not asking them what they think the feedback disc represented. That is a kind of another question, I would say. I think we need to ask them also, what do they think the purpose of the experiment is? What do they think the feedback disc represented? And ask them more questions about what their level of understanding of the study was. So this is sort of what we have right now. The two big claims of unawareness in neurofeedback is this Shibata paper and the Ramot paper. This is the two most cited studies that authors use to argue that neurofeedback can be formed implicitly or without awareness. So I wanted to do my own kind of study. And what I was doing at the time was I was trying to use MEC neurofeedback, so magnetoencephalography neurofeedback, which measures, just like the EEG, measures the, in this case, the magnetic signal that is coming from the brain, trying to get participants to change the activity in their brain related to attention. So attention can be measured over the parietal cortex, and you can measure attention in the alpha frequency. And here what I did was I had participants either train their attention to the left side or train them to the right side. So each time the participant increases their alpha in one hemisphere and their attention moves to one side, they are getting a reward in terms of this GABA patch that becomes more less transparent, or you can see it better. So participants are just asked to get this GABA patch to become clearer, and each time they do, their attention or their alpha is actually changing. So I haven't told them that I'm actually training their attention. I haven't told them of what the purpose of the experiment is. So unbeknownst to them, I'm actually training their attention. And what I was hoping to do was get them to see a multisensory illusion to a greater extent. So I tested them on an illusion, which is called sound-induced flash illusion, which is where you have these two sounds that appear, and then you have a flash. And if you present these in the right way, you can sometimes have participants report that they saw an additional flash, which is not there. And I had the hypothesis that attention would train and change this illusion. So this is the purpose of my experiment. It was to train their attention and to train their multisensory perception to one side, either the left side or the right side. So this is what I was doing in my PhD. So what I then did afterwards was to assess if they knew the purpose of the experiment. Was I did an extensive post-scan questionnaire. So I had participants sit down for 20 minutes to go through this long post-scan questionnaire. And again, I did not know which group they were in. They did not know which group they were in. I didn't know if these participants I was talking with was trained to their attention to the left side or to the right side. And they didn't know if they were trained to the left side or to the right side. So that was all part of a computer program which had randomized them. So I didn't know. And then what I did was I wanted to get the participant to tell me more about their experience. So I had them come into sort of an introspective stance where they could reflect on, or what I would call their pre-reflexive experiences, which is a term used in neurophenomenology, where you ask the participants using their own language. This is because a participant might not even have thought about the experiment. They might not even know what the purpose is, and they just want to get out of the scanner and get home. They've also been part of a pretty weird experiment, and they think that there must be some sort of purpose, and they don't need to know the purpose. And now if they're suddenly asked about the purpose, it's just on point, then they might say, well, I don't know. And they just want to get out of the experiment, right? And they want to get home. And especially if you're doing a very weird experiment like this, where you're viewing black lines that become more clearer, and you're asked to train your brain, and you're seeing all kinds of illusions, it's a pretty weird experiment. So in order to get them to open up, I first asked them sort of what strategy were you using? Were you using an auditory strategy or visual strategy? What were you thinking about inside the scanner? What were you doing? Were you doing anything specifically? And when the participants talked, then I tried to use their language. I tried to make them open up about what they were experiencing as they were doing the training. And then I told them about this illusion, that this illusion, you can actually experience these illusions, and it's totally okay for you to experience these illusions. This is what the brain does. But do you think that you had more or less flashes before the experiment? Do you think in general that you were just having a lot more flashes or less flashes after the neurofeedback? Were you more confident in your response? Do you think that your reaction time, in terms of how fast you were responding to whether or not there were 2 flashes or 1 flash, do you think you were faster or slower? Were you more confident in the way you were doing it before or after neurofeedback? And what this is, these 2 things that I just mentioned, this is a decoy purpose, because this is not what I wanted to do. I did not want them to have more flashes in general, or be faster, be more confident. This is not what I wanted them to do. I wanted them to have more flashes in one side. And then, or I wanted them to have more attention to one side, and therefore more flashes. So this is the actual purpose. Then I also sort of opened them up to this idea. Maybe were you attending more to one side during the neurofeedback? Were you attending more to the left side or to the right side? What do you think? Do you think that your perception of this illusion was changing more in your left side versus the right side from before neurofeedback to after neurofeedback? And I asked them all of these questions, and some of them are decoy, and some of them are the actual thing. And again, remember, I don't know which side. I don't know if it's left or right. They don't know if it's left or right, but they have had an experience. I tell them that they are the experts of their experience, and they should think about it. And then I can kind of say that I've opened them up to these options, you could say. And then what I do is I have this, what I call the confidence token game that I developed. So the participant is given 6 rectangular squares, and then they're shown this sheet of paper. And the sheet of paper has 7 different items, you would say, 7 columns. And this one says experience less flashes in the task overall, experience more flashes, report flashes in the task faster, report flashes in the task slower. Or this one, experience more flashes in the right side and experience more flashes in the left side. And these 2 are the actual options, the actual correct options. And then the feedback was random. So there's also a column that says feedback was random. And in the actual sheet of paper, this is not red or blue. It was also gray. But this is just to indicate here that these were the 2 correct options. And then the participant with their red rectangular squares, they're told that these rectangular squares, they are representing their confidence in what they think the neurofeedback was trying to change about their behavior from before the neurofeedback to after the neurofeedback. And the participant is asked to put these squares where they think that they know, they feel confident that the neurofeedback was trying to change about their behavior. And they're told that they can change these however they want. Like they can put 6 here, they can put 1 here, and they could put 1 in each if they think that it may be all of them. They could put all of them inside the random option if they think that it was just random, the feedback was just random. Or if they put it in here, that's sort of what I think could be or is correct. But again, I don't know which side they were trained. And the participant is given much time, and they're asked to say how sure they are of their placement. And before I even let them put the rectangular squares, I'm telling them that neurofeedback could change all of these behaviors. Neurofeedback can change alpha activity, we know that. So that could change how much flashes you perceive in the task overall. Neurofeedback can also change confidence, so that would change how fast you react. So it could also change this. Neurofeedback can change how you perceive things in different sides, in the left side and the right side. So this one could also be correct, and this one could also be correct. And sometimes in these neurofeedback studies, the feedback is indeed random, and it may be that you are in the random group, and you can place them here. And I tell them, the participant, that I don't know which group you were in. You have to guess. So it's their experience. So this is my opinion, that this is the way that you can ask them these questions in a blinded way, and you can open them up to the experience, and they can sit and think about it where they have the time. And again, they have been warmed up to all of these options, and they're not sort of ready to say if they have some residual degree of awareness. So what I'll show you is this plot here, and the first one here is what they think if they were having their attention to one side or the other during neurofeedback. And here participants' answers were somewhat spread, but in general, they were more on their trained side. So if they were trained to the left, then they were reporting more on the left side. If they were trained to the right, then there was more on the right side, in this trained side. But critically, if we look at this, the contingency, what is the name of this again? The confidence token game. When I look at just have they put things into the left or the right column, then most participants do not put confidence tokens in the left or right column. They say something else. But those that have put confidence tokens into the right or left columns, they have all put them into their trained side. So those who are trained on the right side to have more flashes appear in the right side, they actually put more of these tokens here in the right side. And those people who are trained on the left side, they will put the tokens here in the left side. And these participants are those that I would call those who have awareness of experimental purposes. They've at least put some tokens into that option, and they have some awareness that of the purpose of the experiment. And when I then at the final question ask them, okay, so if this experiment had the purpose of training your attention to left side, to the right side, and you have to answer, do you think we were trying to train it to the left side or to the right side? How much would you say we were trying to change your attention? If you think it was not, then you will put it in the middle. If you think it was left, then put it more to the left. If you think it was right, then put it more to the right. And here participants report those participants who had this degree of awareness, they actually guess correctly to their trained side. So for me, this indicates that a subset of subjects, again, 5 out of 20, they gain a degree of awareness of the experimental intentions, as you can see from their correct guesses about the intentions of the experiment to affect their multisensory perception. And this was in a double-blinded group assignment. I didn't know which group they're in, they didn't know which group they were in. We argue that it is critical to do these types of assessments of the double-blind in neurofeedback studies, and indeed in clinical neurofeedback studies. And we suggest to use this extensive questionnaire to assess this. But it should also be said that this is just one study, and it's only 20 participants and 5 of them, and it needs to be replicated before it really makes sense to do an extensive questionnaire like this, because it takes a long time to do this type of questionnaire. But it presents a way to assess if there's a residual degree of awareness in neurofeedback studies. And the entire questionnaire and all the data and all all the code for analyzing the data is made freely available. So what I will say with this is this shows that it's hard to really say that neurofeedback is performed completely without awareness of the participant. This is also what Timo Kvamme in a way kind of showed in the beginning, as this is the first communication about what neurofeedback is. Conditioned discrimination of your own EEG alpha activity, your own alpha rhythm in humans. This was the first communication about what neurofeedback is. And discrimination, meaning the participant is able to discriminate their own state. I mean, the analogy that I could make is, if I can control my biceps, for example, I can control if my biceps is flexed or not flexed. That is control. And when I learn control of flexing my biceps, you can give me a discrimination task, which is to ask me, okay, is my biceps flexed now? Is it not flexed? And because I'm able to control it, I am also gaining the ability to discriminate it. When you control something, it leads to discrimination. At least that's the case for muscle control, and it seems to be the case for alpha activity as well. And this is something that is also shown in a paper by John Frederick. And so John Frederick has replicated many of the findings from Kvamme from the 60s. And this conclusion that it's hard to delineate implicit from explicit components of the neurofeedback learning, and it's not trivial, and it requires some more theoretical and methodological discussion. This is the sort of a point made in this neuroscience and biobehavioral review paper that is a really good review of this whole problem. And it also says this, that in humans, the bulk evidence supports the view that learning is often accompanied by contingency awareness. So in that sense, it isn't surprising that that is what we also find in our studies. So why is it that my study finds one thing and Shibata and Ramot find something else? One thing that I've come to consider, of course, is that we're training different brain activities. They are training visual cortex or higher order visual areas, or they're training fusiform face area and parahippocampal place area, these higher order visual regions. And I'm training alpha, and it may be that alpha is something that when you train alpha, you become conscious of it, but when you train these other areas, you don't. So again, they use this decoded fMRI neurofeedback of these low visual to high visual areas, and maybe when you train these low and high visual areas, it doesn't become conscious, and it is only the information processing between those areas are only very local and sort of locally confined in those areas, whereas alpha is a more global signal that is perhaps reverberated, you could say, into a global neuronal workspace where it becomes conscious, you have conscious access. So this is the way I describe it using the global neuronal workspace theory of consciousness, that perhaps this is an explanation why we find there's awareness, and when you train these other areas, you don't. One thing that relates to this is this type of what is called information transmission analysis. And here in the paper I mentioned before, where you have this snake that is being, the representation of the snake that is being trained, what you can do is you can ask, so the other brain areas, the brain areas that are not trained in the whole brain, do they predict the activity in the trained area? So you're training these areas involved in the visual representation of the snake, but other areas of the brain, in the frontal part of the brain, the parietal part, and so on, can they predict the change that is occurring in the targeted brain region? And what is shown in this paper is that when you consciously see the actual snake, when you see the real stimuli, other areas of the brain are able to predict, or they're better able to predict this snake or the activations in the targeted area. Whereas doing unconscious neurofeedback, doing this kind of neurofeedback, these other areas of the brain are not able to, or to a less extent able to predict the representation in the targeted brain region, in the region that's undergoing neurofeedback. And that is at least a type of evidence, you could say, that the activity is local and it's confined within one area, and you're only training that one area. And that is at least some part of an evidence for the claim that neurofeedback is performed unconsciously. And this is a new type of approach, new type of machine learning approach to sort of get at this. But what I will also say is that I still want to ask this question: if participants have a residual degree of awareness during experimental purposes in neurofeedback, and is the double blind possible in clinical neurofeedback studies? It's still a big question. The Shibata and the Ramot paper have some tests of awareness in their article, but I would say that they're not exhaustive tests of awareness, and that what I have done in my study is more detailed and also took more time, really get to know my participant and really get to ask them what their experience was of what they were experiencing, and thereby figure out if they had awareness. And then there is, of course, this use of information transmission analysis, which is performed in some of these decoded neurofeedback analyses. And I think the next step is to try to link these tests of awareness with this information transmission analysis. That's at least one way forward. And then, of course, it's important to keep in mind which brain area is being trained, or brain pattern is being trained, or dynamic is being trained. And training alpha might be different from training a locally confined area in the visual cortex, for example. So that's important to keep in mind in the future. But I think it's still sort of a question to ask if there is a residual degree of awareness of the purpose of the experiment. Yeah. So that is it. And yeah, thank you for your attention, and also special thanks to my supervisors and to everyone involved in these studies that helped make it possible.